Welcome to Hibberd Health. I feel very lucky and honored to have Dr. David Scheim join me today. Dr. Scheim, thank you so much for being here today.
It's a pleasure to be here with you.
Thank you very much. Dr. Scheim was awarded a PhD degree in math from MIT and then completed postdoctoral fellowship in mathematical biology at the Los Alamos Scientific Laboratory in New Mexico, USA. He served as a commissioned officer in the U.S. Public Health Service at the National Institute of Health for 20 years. He continued work as a contractor for the NIH on medical database development, and his independent medical research shifted focus to therapeutics for COVID-19 in 2020. He amazingly is also an author of a #6 New York Times nonfiction bestseller on the U.S. history, unrelated to science. Dr. Scheim, you wrote a recent scientific preprint titled From Cold to Killer, How SARS-CoV-2 Evolved Without Hemagglutinate and Esterase to Agglutinate. Then clot blood cells in pulmonary and systemic microvasculature. This is profound information. Can you please elaborate on the key points in your paper?
Sure. Well, number one, let's just start from the fact that as clinicians have studied more and more cases of COVID-19 over recent months, they found that vasculature occlusion, that's blood clots, is central to COVID-19. Now, it turns out that the SARS-CoV-2 virus is part of a family of coronaviruses, and especially, in particular, beta coronaviruses. And that close relatives of the SARS virus, SARS-CoV-2, are common cold viruses. And somehow those pass through, they're pretty much harmless for everybody. But COVID is deadly. And a key difference is certain properties of the SARS-CoV-2 virus that make it more prone to agglutinate red blood cells. So that's where the focus of this paper lies, on the biology of the virus that makes it susceptible to clumping red blood cells. And it turns out that this is the cause not only of things like COVID toes or clots in assorted organs, but it actually seems to be the very basis of the damage in the lungs. Because it was first thought, oh, the lung tissue is damaged, the patients are having trouble breathing. But in fact, when they look closely at the lungs, in many cases, it's the vasculature in the lungs that's damaged, and the actual lung cells, the alveoli, are fine. At the same time, it's very well known that with COVID patients, their lung mechanics are fine. They can breathe in and breathe out. It's just that the blood cells are not getting oxygenated. So it's really the blood that's the key to all this, and it's the biochemistry of the SARS-CoV-2 virus that's at the heart of all of this.
It's really interesting. Now I'm going to go back to your prior preprint of July 2020, which focused on ivermectin as a promising treatment for COVID-19. Does your current paper also cover developments for ivermectin?
Absolutely. I think there's a lot of background, but the bottom line is whatever theory we find, we want to be able to apply it towards a remedy. And the bottom line, this is at the end of the paper, I review the latest clinical trials. There's 8 of them reviewed. One is a randomized controlled trial for prophylaxis. And there's one more that recently came out that's a randomized controlled trial for treatment that's not in the paper. But in any case, the evidence is accruing study by study that ivermectin is a highly effective treatment. The thing is, though, we need to understand why it works. If we have these clinical results and there's really just scattered theories that are very compromised as to what's going on, it holds back the development of ivermectin. So this paper lays a foundation for looking at some credible theories of why ivermectin works. And in particular, in order to understand the biology of COVID-19 and how ivermectin works, we really need to understand basic principles of Virology 101. These were well known to Jonas Salk in his study of viruses, but somehow in this age of molecular genetics, some very basic principles of how viruses work have been forgotten, and it's very important for us to know that.
Yes, I could appreciate that. Would you like to elaborate on what these principles of Virology 101 are and that are being largely ignored today in COVID-19 research?
Yeah, well, way back in the 40s, there was an assay called the Hemagglutination assay, in which you take some viruses and you mix them with red blood cells, either human or animal, and lo and behold, you get this interlaced sheet of viruses and red blood cells, and sort of you can just see it floating on the top of the little culture well. And it was well known, and this assay has been repeated decade after decade. It's just been less important with genetic tests available to assay viruses, but it's well known, still used. And a very interesting additional fact is when you get these interlaced sheets and you wait like 15 to 20 more minutes, the sheet collapses amazingly. It's a pretty graphic kind of image of what happens. So the reason for that is the viruses have certain little attached places of attachment where they attach to red blood cells, but the viruses also paradoxically release an enzyme that dissolves the receptor in the red blood cell to which the virus attaches. And it turns out viruses need to navigate this subtle balance of being able to attach to cells, but also release from cells, because there are certain kinds of attachments that if the virus stuck there, they could never infect the host. So it's a complex balancing act, and I don't want to go too much into all of the nuances in my paper, but it is very important that many, many strains of viruses attach to red blood cells.
That's really interesting. Thank you for sharing that. Do we know that the SARS-CoV-2 virus in particular binds to red blood cells?
Yes, we absolutely do. And one thing, when I started my paper, I realized that there were certain basic principles that I was laying out. And if I was not absolutely 100% sure and did not have the studies that proved it, then everything else would be tenuous. So I really spent some weeks looking carefully at the studies. And in fact, there is one. There's something called the heme absorption assay, which is almost identical to the hemagglutination assay. And there's one study where they took SARS-CoV-2 and used that assay, and lo and behold, it bound red blood cells, this virus. But more important, clinically, which is quite amazing, and I'll come back to this later, blood was drawn from several COVID patients, and the red blood cells from these COVID patients, 40% of them had viral traces. So they were at some point bound to virus, and it looks like the virus may have broken off from the cells. You have both cell culture and clinical evidence the SARS-CoV-2 virus binds to red blood cells.
That's interesting. So which receptor is key to SARS-CoV-2 binding to the red blood cells?
It's actually something called sialic acid. And sialic acid, there are different types of receptors, and typically this little sialic acid molecule is right at the tip of a receptor. But this is actually what binds to a region in the viral spike protein of the COVID-19 virus. And as you've probably seen the little pictures, the virus has some 100 little spikes, and that's where the spike protein is. So it's the sialic acid. It's a kind of a sugar molecule, and there are different variants depending actually in different animal species and in humans. But so this, the SARS-CoV-2 virus binds to the sialic acid molecule, and this is so important because it is well known by everybody, all investigators of this virus, that the way it replicates is it works its way to an ACE2 receptor. And the virus fuses with that receptor, it slips in, and it replicates through that receptor. However, the number of ACE2 receptors in cells is much, much less than the number of sialic acid receptors. And actually, when the virus first approaches a cell, it attaches to a sialic acid receptor. Then eventually, and that's typically a weak binding, and until there's ways that it's strengthened actually by multivalent bindings, but it attaches to one of these, and then it works its way and slips over to an ACE2 receptor. But it's the whole initial attachment to sialic acid is very, very important in the whole process of infection of this virus.
So to double check for our audience listening, although in your paper you state many references about viral bindings to sialic acid receptors, but do we know that this applies to SARS-CoV-2?
Yeah, this is another thing, as I said before, if I'm going to base my whole paper on this, I needed to be absolutely sure. And there are several really good studies that talk about beta coronaviruses and how they bind to sialic acid receptors. So the biology is well known and described in detail. But to confirm that this actually applies to SARS-CoV-2, the best study is one in which an investigator made a nanoarray that's using nanotechnology very closely spaced together of sialic acid molecules, and the virus bound very well to these nanoarrays of sialic acid. And in fact, this whole system turns out to be a very sensitive method of detecting low concentrations of virus. So it is absolutely confirmed. And this is what happens in host cells, that the sialic acid is very densely distributed. So it serves an effect as a nanoarray of sialic acid molecules. So again, there's a lot of studies about viruses that are in the same family as SARS-CoV-2, but there's this very good study for this very virus.
That's interesting. So how does sialic acid figure into the body's primitive cellular defense against viral infection?
Well, the body is loaded with sialic acid molecules. Like in your mucus, there are mucins, and mucins is nothing but tons and tons, like billions of sialic acid molecules. But your red blood cells we have trillions of red blood cells, and each red blood cell has millions of sialic acid molecules, very densely distributed. And they're actually arrayed on a kind of a molecule that has no biological purpose whatsoever that anyone could see, other than really serving as a decoy. The virus comes in, and instead of getting to go right to a host cell, it's presented with all this junk, sialic acid, mucus, and red blood cells all over the place. And that evolved so that the body can blunt this viral attack and sort of act as decoys for the virus until the body has a chance to build up a specific antibody system. The only problem, there's a little bit of a twist to this. In its attempt to protect the host cells, the red blood cells bind so well to the viruses that they clump. And this creates its own other set of problems. And this is where we find that a subtle difference in the biology of the SARS-CoV-2 versus the related common cold virus allows the SARS-CoV-2 virus to bind more tightly to the red blood cells. And this is where the problems are created when you have these clumps. And it's not just to red blood cells, it's also to other blood cells and to endothelial cells. These are cells that line all the blood vessels, and viruses bind to those as well. What happens is you get this clumping, and the clumping starts obstructing the capillaries. and then the blood flow slows down, and then it's easier for the virus and the blood cells to clump with less blood flow, slower, then there's less forces to break up the clump. So it becomes a cascade. So that's where this primitive defense mechanism just comes into play, but it becomes counterproductive for SARS-CoV-2.
Very interesting. What evidence do we have that these clumps actually form clinically during a COVID-19 infection?
Well, this paper cites a number of studies. The first one, and let me repeat this, is a study looking at the red blood cells from several COVID patients, and they find that 40% of the red blood cells have traces of the SARS-CoV-2 virus on the red blood cells. So that's a pretty high percentage, and it looks like this suggests that the virus binds to the red blood cells, but as it's all flowing through the blood with a lot of force, the virus can just break off and leave a little trace in it. But we also look at the capillaries throughout the body, like you can either look at things like COVID toes or look at even the lungs, and you find there's a virus lining the capillaries are damaged, and when you look at the lungs, you find cases where the capillaries seem to be blown apart and red blood cells shoot out through the capillaries. And what it looks like is that these clumps of red blood cells and viruses and endothelial cells can form obstructions, but there's such force coming from the pumping of the heart that it could just blow these apart. And we find in many patients that it's the capillaries that are damaged. The alveoli look fine. They look pretty normal. The breathing is fine, but the capillaries, it's the transfer of oxygen to the blood cells that is off. So my paper goes through a lot of these, and there's some really nice figures. Let's see. Let me see if I can pop one up here. Shares.
Yeah, that'd be great.
Here, this is from the lungs, and you see the red blood cells. They're very much damaged. They're shot out. They just break through the capillary wall. And this could be explained by clumps of these red blood cells and viruses under the force of a pulse from the heart of just being shot through the capillary walls.
That's a great picture you showed us. Thank you very much. Now, are there any other diseases in which such clumping of blood cells plays a key role in its critical symptoms?
Yes, it's severe malaria. In that case, the malaria parasite gets into the red blood cell and it acts sort of like the virus. The malaria-infested red blood cell tends to clump to other red blood cells and also to endothelial cells. And in this case, it's been studied very, very carefully, and it is absolutely confirmed that the real serious problems in severe malaria are caused by this clumping. It's exactly what I've been proposing for COVID-2, that we know that this happens in malaria.
Very interesting. Are there any other clinical findings that support the blood cell clumping that you describe as a key cause of the morbidities of COVID-19?
Yes, there's some very interesting studies conducted by a group headed by a pulmonologist and an engineer that developed some advanced image analysis processing for lung CT scans from patients. So when they use this analysis on COVID patients, they find that the blood flow is redistributed from the smaller capillaries in the lungs to the larger vessels. And they also find that when they look at the breathing function of individual patients, the more disturbed the breathing, the greater the redistribution of blood flow from the smaller to the larger capillaries. So this really is consistent with the finding of this paper that it's this clumping that is the first cause of the breathing function.
Yes, wow. So, how does the cytokine storm and hyperimmune damage fit into your explanation of virally mediated clumping of blood cells being the prime cause of COVID-19 morbidities?
Well, a lot of the histological studies of COVID-19 patients are done from autopsies, and these are from obviously very severely ill patients. And if you start with these clumps and then you go on for weeks, eventually clotting mechanisms kick in. You have fibrin threads that sort of interweave, and at that point, the little clumps, they're no longer reversible, they're hardened. So there's no question that when you look a lot, especially at severe patients, there's a progression called the coagulation cascade, where just this blood clumping hardens, and then various immune cells kick in, and that's the coagulation cascade. In the early stages, it's more reversible, and we're going to get into ivermectin, I guess, soon. But what's stunning is that classic for ivermectin is you have a patient with declining breathing function. In some cases, by the hour, they're losing breathing function. They're not yet in the ICU. They're still at home or on a little bit of oxygen. And then they take ivermectin, and the next day they're stabilized and back to normal in another day. That's very classic. That certainly means we do not have a clot. Clots do not reverse in a day. Clots take weeks to reverse. It looks like what we have is a clump. So it's very important to note there's what appears to be a progression of this reversible clump, virally mediated clumping, that if you wait long enough, then you get this immune cascade and the clotting.
So, how does the role of virally mediated clumping of blood cells in COVID-19 relate to known incident factors for this disease?
Well, what the most striking difference in the course of COVID-19 that we know of occurs between younger and older people. Generally, younger people are barely symptomatic. It's sort of like a cold for them. Older people are the ones who have the severe morbidities and much higher death rates. It turns out that in younger people, the blood flow in certain capillaries is twice as fast as that in older people. In other capillaries or arteries or veins, it's not quite twice as fast, but it's much faster. So here you have this blood flowing at a vigorous rate in young people. So yeah, you have the virus, it comes in, you have the blood cells, they sort of attach, but boom, they're flowing, twisting, turning quickly, and they never get traction. They bounce around, but you never get these clumps really starting to form. Whereas in older people, where it's slower, then these clumps can start to form, and then in a cascade, slow down and down and down, more clumps. So that's one striking incidence factor that that fits this theory. Also, it turns out, although generally we think low blood pressure is healthier, it turns out that in COVID, a systolic pressure less than 90 millimeters is associated with a significantly higher incidence of COVID. And conversely, if you sort of factor out for other risk factors, people with higher blood pressure have less mortality with COVID. And that fits the same pattern that the more the blood is turbulent, the less chance there is for these clumps to form. There's been a few really good studies on blood type, and type A and B blood tend to be more sticky. Under normal conditions, it doesn't really matter. The blood doesn't clump. But if you have a situation where there's other clumping factors, type A and B tend to be more agglutinating. And it turns out that type A and B blood are associated with higher, I think in some studies, higher incidence and other higher mortality than they are versus, let's say, type O blood. And interestingly, you get exactly the same pattern in severe malaria. Same deal, more clumping, and then you have the clumping that's caused already by the malaria parasite, so you compound it with type A and B blood. And there the effect is very striking, and people even think there's an evolutionary preference for type O in certain regions to avoid greater risks of malaria.
That's interesting. So I take it that AB blood is a double whammy. Am I right?
Yeah, yep.
And type O blood, so for malaria, people with type O blood are showing more resistance?
Yeah, that's correct. Yeah.
Very interesting. Wow. Now, how does the insight into virally mediated blood cell clumping in COVID-19 relate to the effects of ivermectin in this disease?
Well, we have many clinical studies I review, but we have a striking pattern of clinical benefit. But one particular pattern, I'm just going to recapitulate this. I mentioned it earlier in the Florida study. And I have a good friend in Atlanta, and her case really got me interested in ivermectin, but she epitomized this pattern found in the Florida study where a patient has COVID, they may be doing okay for several days, as is typical, and then all of a sudden, a decline in breathing function that can get scary, where even you just sitting still and having trouble breathing. And then they take ivermectin, and for my friend, and typical of the Florida cases, by the end of the day for her, she was stabilized. The next day, she could start to walk again, a couple of days back to normal. So ivermectin, one logical explanation is ivermectin is slipping in and preventing this clumping of viruses and blood cells. And there are 6 different molecular modeling studies that look at sort of how ivermectin would logically bind to regions in the viral spike protein. And all 6 of them found that ivermectin comes out on top. And these study, most of these weren't looking at ivermectin at all. One study said, let's look at 100 molecules that could have benefit for COVID, and let's see how they would bind to regions in the spike protein. And just blindly out of 100, ivermectin comes out on top as the most efficient blocking molecule. So given all these studies, although it doesn't prove it, it makes a logical case that ivermectin slips in, and as these clumps are sort of bouncing around and forming and unforming, ivermectin slips in and just loosens the clumps. I think there's a pretty reasonable case that that's a mechanism of ivermectin. So what we find now, I will say there are some other studies that were done way back at the beginning of COVID. It was a cell culture study. They used more than 1,000-fold the concentration you would ever get in tissue, and they found that ivermectin was an effective antiviral for the COVID virus. Also, the mechanism that they proposed from that study did not apply to an RNA virus. It talked about changing the dynamics of chemicals in the nucleus, but that doesn't typically apply to an RNA virus. And I think this was great work. It aroused a lot of interest. I think a lot of scientific discoveries sort of piggyback, and you start with one and then you find, well, it's not quite right, and you go on from there. I'm not faulting these people, but it really does hold back the development of ivermectin when a serious scientist looks and sees someone cites a mechanism that makes no sense. That's just not supported. And I think blocking the viral spike protein is reasonable. But what we do find, if you focus on that mechanism, is that this does apply at clinically achieved concentrations. It's important to realize, though, that there's a standard dose of ivermectin. Like if you have scabies, the standard dose is 200 micrograms per kilogram. That might be about 12 milligrams for an average weight person. And that's fine for scabies, but it does seem that for COVID, that there's a dose response effect, like there is for a lot of drugs. The more dose you get, the better you do against the disease. It turns out that that ivermectin can be given at 10 times the standard dose without a bit of harm. There's studies, it's not even that this is a maximum, it's almost that you're fine at 10 times the dose. And it looks like when you start looking at the clinical studies, the published studies, but also the anecdotal evidence from really credible doctors as they're reporting it, it really does look like the higher the dose, the better people do. So if you start looking at a credible mechanism, it helps you more to start evaluating what's the optimal dose, what's really going on. I think there's a lot of advantage to digging in and finding a solid scientific foundation for the action of ivermectin in COVID.
So putting all of these findings together and including what you've just been talking about, what do you see as a way forward with ivermectin for COVID-19 treatment?
Well, I think that more and more clinicians are using ivermectin. I don't think we need randomized clinical trials. In fact, I think they're unethical. When you're at a point where ivermectin is going to save 3/4 of the patients, it's just not ethical to send some into a control arm. I think what we need to do is look at the published studies and the emerging data, try to find the very best dosing, the very best scheduling of dosing, and the very best adjuncts. And it's well known that using ivermectin in conjunction with the antibiotic doxycycline, which is also antiviral, that really does improve results. There's some indications that zinc adds some, and whether it's a 20% benefit or whatever, if it consistently improves performance, you want to have another adjunct. And then you also want to give this as soon as you have an indication from symptoms that you have COVID. Because again, we talked about how clumps can turn into clots. And what you want to do is prevent this virally mediated blood cell clumping from rolling along before it rolls along too far. You don't want to wait for the breathing crisis to happen. Mm-hmm. So what would be great if we had, in countries where there's better testing, the best thing to do, you go a couple of days, you're pretty sure you have COVID, go get a test right away. And even if the results take more than a day or two, start taking the ivermectin. I think that's how a lot of clinicians are doing it. It'd be great for research purposes to get that test. If you're in the US testing right now is just so it's a joke. You have to wait days in a lot of places to even get a test. But if you're in a country with the same policy, you get the test. If you're pretty sure, don't wait for the results, start taking the ivermectin. And it really does seem like the whole tenor of the disease changes from a threatening serious disease to something you're probably just going to ride out with minor symptoms. That's the sense I'm getting from the best doctors who are using this.
That's really great to hear. Now, what about the role of vitamin D and vitamin C as adjuncts?
Yeah, I think there's good evidence for that too. And I think we just have to look at what the best doctors are doing and who's getting the best results. And there seem to be a lot of adjuncts like that would make sense to take.
Yeah, there's a few others mentioned, melatonin, aspirin, when you're getting further into the disease.
Now, where we look at the evidence, I don't think we can conclude that 100% of patients who take ivermectin at the right time, that every single one is going to come out with, just improving and not getting worse. I think that what we're seeing is suggestions that a large majority are fine, but if any find that they're worsening, then there are some other things that are being found effective for later stages, like dexamethasone and even high and low molecular weight heparin seems to be effective, and that may not only have an anti-clotting effect, there's indications that it actually also may be a masking molecule for viral binding to host cell receptors.
Yeah, well, that makes sense because every individual is different. And most certainly there are different things going on in the body that can affect their interaction with a drug. That's why under many diseases, there's alternative treatments because not everybody responds. to the same drug, however ideal it may seem, to treat that disease.
Right.
So ivermectin seems to stand out as having more effective response to it than any of us have seen with any other drug.
Yeah, I agree.
It's really interesting. Very interesting. Thank you so much for this. It's been a really interesting talk. I really appreciate you being here with me today, and I hope we can continue our discussions and go into more detail about all this phenomenal information.
Well, it's been my pleasure talking to you, and I look forward to seeing more research emerge in the days and weeks ahead.
Yes. Thank you very much.
Well, thank you.
For being with me. Thank you. And thank you everyone for joining me on Hibberd's Health. Please subscribe to my YouTube channel so you can see new videos as they get uploaded on a regular basis. Thank you very much.